Hello, everyone. My name is Hechen Li, a second-year PhD student from Georgia Tech. And today I'm going to introduce scMultiSim, simulation of multi-modality single-cell data guided by cell-cell interactions and gene regulatory networks. In recent years, we have seen a lot of new computational methods that reveal information on single-cell data. For example, using the gene expression data, we can infer the gene regulatory network, or GRN, inside cells, and the cell-cell interactions, or CCI, between the cells. Using the splice count and the unsplice count, we can estimate the RNA velocity and so on. However, there is too little ground truth in the experimental data. Like, if we don't know the real GRN in the data, how do we know our GRN inference method is correct? Therefore, we have simulators that generate simulated single-cell data from known ground truths. They are widely used to help us develop and benchmark new computational methods. Current simulators usually focus on one biological factor. For example, a simulator may produce single-cell RNA-seq data affected by a GRN. However, in our cells, there are lots of different biological factors that affect the gene expressions together, such as GRN, cell-cell interactions, chromatin accessibility, and so on. And moreover, recent computational methods are utilizing multiple data modalities. For example, they jointly use single-cell RNA-seq and single-cell ATAC-seq to perform data integration and get better performance than previous method that only use one single modality, like single-cell RNA-seq data. Therefore, we need a simulator that can simulate multiple biological factors and also output multiple data modalities. And that's why we designed a simulator called scMultiSim. As shown in section 2 of the poster, the minimal input to scMultiSim is just a cell differential tree that controls the cell population and an optional GRN. So what kind of biological factors can be simulated in scMultiSim? It can simulate the effect of chromatin accessibility, GRN, and spatial cell-cell interaction simultaneously. And what kind of data modalities can be outputted? It generates continuous or discrete cell population and outputs single-cell RNA-seq data, including the unspliced count, single-cell ATAC-seq data, cell metadata such as the pseudotime, spatial cell locations, and the ground truth of RNA velocity, GRN, and cell-cell interactions. It can also add technical noise to data such as batch effects. So scMultiSim has three major highlights. The first one is it generates data with realistic complexity, so we can test the real-world performance of a method. And the second is using all these outputs, we can benchmark a wide range of computational methods, including trajectory inference, GRN inference, CCI inference, RNA velocity estimation, multimodal data integration, and of course, clustering, and so on. And the third is the data modalities in the outputs, namely the single-cell RNA-seq and single-cell ATAC-seq, they are coupled or correlated. So we can use them to benchmark methods that jointly utilize multimodal data. Section 3 of our poster briefly introduced the main concepts of the model, and you're welcome to read our paper for the method details. And looking at the section 4, the results, in figure A, we show that scMultiSim can simulate continuous cell trajectory or discrete cell clusters. And users can easily control the population using parameters. And in figure B, we also validate that all the biological effects are successfully simulated in the output data. We show that the genes regulated by the same transcription factor have higher correlations between each other, and we also get higher correlations between cells with cell-cell interactions. And finally, in figure C, D, E, F, we demonstrate scMultiSim's potential by benchmarking 4 challenging computational tasks: GRN inference, RNA velocity estimation, multimodal data integration, and CCI inference using spatial data. Notably, for the first 2 tasks, we got the results that are in line with previous benchmarks. And finally, we believe that scMultiSim could be a powerful tool for fostering new computational methods for single-cell multi-omics data. And with more subsequent benchmarks, it can also help researchers choose a proper tool based on what kind of data they have.